So let's talk about some other genome assemblers. So ALLPATHS-LG is another one which is kind of more successful approach. What are genome assemblers? Genome assemblers are variants of simple sequence alignment programs. We'll talk about sequence alignments in some later sections. So after our genome is assembled, then we will try to compare those parts of those genomes to other genomes. So that is where we have those sequence comparisons. So in that sections, we'll talk about those alignment programs. So assemblers are special kind of alignment programs which are there to align those short sequencing reads and assemble the genomes. This ALLPATHS-LG is a short read genome assembler from the Computational Research and Development Group at Broad Institute. So remember, we talk about Broad Institute regarding the generation of high throughput data in some previous sections. So that institute also has this genome assembler put together. The goal of this assembler is to generate high quality genome assemblies using short reads, approximately 100 base pairs, which are mainly coming from machines like Illumina, and it can put them together. So what it does is that assemblies are not linear, so it's not like it's putting them in a linear fashions. So it's presenting those assemblies in the shape of those graphs. So I will look into this algorithm later on. So ALLPATHS sequencing model, what we can do here is in this comparison, we can see that how much coverage we can use. So we have the libraries and insert types. So we can have fragment libraries, just simple single end reads, no paired end reads, just fragments. Their size is 180 base pairs. And the reads what we can generate from here is around slightly over even 100 base pairs. And for this kind of assemblies, if we are using those simple single reads, we might need a coverage up to 45x in order to use in this ALLPATHS-LG assembler. If we have short jumps where we can have the fragments, the insert size is small, and obviously the reads are 100 base pairs if you are using Illumina. Again, we can have we might wanna have a good coverage for that, so that is 45x. Long jump, or if we have long insert sizes, the fragment size may be up to 6000 base pairs. Again, the read size is similar, but here we can compromise on even low coverage up to 5x. And phosmid jumps, where we are using large, huge fragments, where we have 40,000 large fragments. The read lengths are smaller, and for that, if we are using this kind of libraries, even we can do that once. So 1x coverage is okay for that. So in order to use ALLPATHS-LG, these are some specifications where you can fill them up before you use this software. What are computational requirements for this program? So it needs 64-bit Linux memory, about 160 bytes per genome base, implying advantages. So this assembler is relatively fast, and we can use long reads. And if our genomes are small, so we can use them. So what is ALLPATHS-LG? In this section, we have seen that it is a short read genome assembler from Computational Research and Development Group at Broad Institutes, and we can use it with different coverages as we have seen in this section.